Good afternoon, everyone. Today we look into one of the most transformative techniques in modern biotechnology, which is called enzyme immobilization. This powerful approach has redefined how we harness biological enzymes, offering enhanced stability, reusability, precision in industrial and pharmaceutical processes. So as we look at the principle and applications, you will see how immobilized enzymes are not just tools for convenience, but cornerstones of innovation in sustainable and scalable biocatalysts. Immobilization of enzyme refers to the techniques of binding an enzyme to a solid support, such as inert surfaces or some other matrix, which allows the enzyme to retain its activity during use. And sometimes when you use it in harsh condition, it makes the enzyme to remain stable and also allow it to be separated easily. The process of immobilization of enzyme has several applications in the industrial microbiology, such as food and beverages, production of pharmaceuticals, biofuel, and other industrial production processes. It involves the use of enzyme in a solid support, like I said earlier, and the enzyme and substrates react together, giving us the desired products while the enzyme remain on the solid support or in a matrix. At the end of the day, we get the product out and the enzyme is reused for other industrial processes. So this is a diagram showing us industrial process beginning from the harvesting of cell to the final product. Every industrial bioprocess is usually catalyzed by specific enzyme. So the recovery of this enzyme molecule during downstream process and improving operational stability is highly crucial and is usually expensive. However, when we improve recovery, improving recovery and operational stabilities, it will reduce the costs and also higher overall bioprocessing efficiency. To achieve this, we have to use immobilization technique. So this technique became one of the most innovative and recently well-studied systems to carry out a production on industrial scale with higher productivity and less loss due to other recovery processes of the enzyme that are used, because there's no way you want to carry out a large industrial process without biocatalyst, which can help to speed up the uh, production processes. So the reason why we use this immobilized enzyme is because of easy recovery. Immobilized enzyme can easily be recovered, separate and separated from the final product, and it makes the process to be cleaner and more efficient, leading to reduction in purification processes in the cases where we have to separate the enzyme from the product. And also, this enzyme remains stable over a long time, giving them a longer shelf life and reducing the need for frequent replacement of this enzyme for same processes. And it also has a minimal purification required. Like I said, there's little or no purification that is required because they don't mix with the product. Therefore, there's less need for downstream processes, saving time and preserving product quality. Then it supports continuous processing because the system can run non-stop without interruption or alteration, boosting productivity, consistency, and time. Then enhances stability. Immobilized enzymes are more resistant to heat and pH changes. They prevent denaturation and ensure reliable performance even under tougher conditions. It is very important that we use this immobilization technique, and one of the significance is the fact that immobilization system significantly enhance bioprocess efficiency by addressing key limitations in terms of all the restrictions that are usually that serve like hurdles in production process. So they enable the recovery and reuse of enzyme, improve molecular stability, activity, and selectivity, and also increase resistance to inhibitors. This system also streamlines production by eliminating unnecessary separation and purification steps, like I said, ultimately leading to more efficient and cost-effective generation of desired products. At the end of the day, when we use immobilization technique, we discover that our production is faster, then we have a more cleaner and more purified production. And one of the advantages is, like I said earlier, easy separation of enzymes, reduced cost of downstream processing, multiple use of enzyme in terms of recycling. You can use the same enzyme over and over again. They are more stable, and the use of fixed bed or batch reactor without the need of membrane to isolate enzyme from products. And co-immobilization with other enzymes is also possible in the case of immobilization technique using enzyme. However, wherever there is advantage, there's always some disadvantages or limitation in the use of immobilization technique with enzyme. So here are the disadvantages of enzyme immobilization technique. We have sometimes we have lower enzyme activity compared to native enzyme. Sometimes the enzyme can give a lower activity, and there's also additional cost for carrier and immobilization. Then we also have lower reaction rate compared to native enzymes, and sometimes they are subjected to fouling. Then disposal of the exhausted immobilized enzyme in terms of incineration can also be costly, and due to expertise and also the stages involved, this thing can be laborious and time consuming in immobilization processes. However, looking at the advantages and disadvantages, it is better to use immobilization technique because it provides more faster processes, purified products, and it also gives a quality product that is free from contamination with the enzyme, which also requires purification at the end of the day. So we have different types of immobilization techniques. Here we have the adsorption, we have the covalent, we have the cross-linking, we have the entrapment, and so on. So we are going to look at these techniques one after the other, then we'll finish the class. These reactions have a typical types of application. In this diagram, it shows us three common types of immobilization enzyme reactors used in bioprocesses. The first is a packed bed reactor. In the structure, immobilized enzymes are fixed on a solid support, tightly packed inside the column, and it operates by allowing fluid to enter from the top. You can see. Let me get a laser. Okay, you can see allow fluid to enter from the top and flow down through the beds. Now, the advantage in this process is that it allows high enzyme loading capacity, and also is very suitable for continuous operation process whereby the substrates are continuously loaded from the top, and it ensures minimal enzyme washout. However, there are some challenging limitations in the sense that there could be channeling in terms of flow distribution, which can be very poor. Then also it can build up pressure. The second one, which is fluidized bed reactor, in this aspect, the enzymes are suspended in the upward fluid flow, causing them to fluidize and move freely. They are scattered and move freely. The way it operates is that fluid enters from the bottom. It enters from the bottom and exits at the top, keeping particles in clear motion. One of the advantages of this reactor bed is that it reduces mass transfer limitation due to particle movement. Then also, it also lower pressure compared to packed bed, and it is suitable for handling viscous fluids. These are advantages over the fixed bed reactor. Its limitation its limitation includes it requires careful control. of fluid velocity to maintain fluidization without washing out particles. And then the third one, which is the stirred reactor, which is stirred tank reactor. The immobilized enzyme beads are usually suspended in a liquid phase, and it's been operated in a mechanical stirring which ensure uniform mixing. It could also be electrical stirring, which ensure mixing of the substrate and enzyme particles, in an homogeneous way. The advantage of this process is that it allows good mixing and the temperature are well controlled. It can easily scale it up and it can handle varying viscosity of substrate irrespective. However, it also has its own little drawback in the sense that in terms of using it, if it's mechanical stirring, it could damage the enzyme support, or sometimes there could be more complex separation of beads after the reaction. So these are typical reactors that are used for biocatalysis. So for the techniques, the first technique is physical adsorption, which is a method of enzyme immobilization where enzymes are non-covalently bound to the surface of a solid support. The attachment relies on a weak interaction such as van der Waals forces, hydrogen bonds, and perhaps ionic bonding. So it's usually work when you use some common support like silica gel, calcium phosphate, or glass. Sometimes you can use some cellulose derivative also, Cefadex, and so on. It is simple and cost effective with no need for complex chemical or procedure. Some of the disadvantage of this process is the fact that it has a weak binding, so enzymes can easily detach themselves due to changes in pH or temperature or ionic strength. So enzyme can easily detach itself, so this reduces the activity because of the weak binding of the covalent, the adsorption of those enzymes which are covalently bound together. It is used usually for biosensor and bioreactor and diagnosis tools. Let's look at the second method, which is encapsulation and entrapment. The encapsulation process is based on the entrapment of biomolecules in a polymetric matrix. It involves enclosing enzyme with a porous matrix that allows substrates and product to diffuse in and out, but then it retains the enzyme inside the mixture. The enzyme is retained, but the substrate can diffuse in and out. So in this process, the enzymes are physically trapped, but not chemically bound. They are not chemically bound, but they are physically trapped. That's what we call it, entrapment. So advantage of this process is that it protects enzyme from some very harsh environment during the process, and it maintains enzyme structure and activity, and is usually suitable for continuous flow system because the enzymes are trapped. They are not actually chemically binded. However, the advantage is one of the main advantages of the fact that there's a limitation. It can reduce the reaction rate. Enzyme may also leak out over time, and the matrix can be complex. For encapsulation, it involves a closed enzyme with semi-permeable membrane or microcapsules. The enzymes are isolated inside a capsule, and the capsule can be made of liposome polymer membranes or some gel matrix such as salt gel. Advantages of this particular technique is that it provides high protection from external conditions and also allow control and release a reaction. It is usually ideal for sensitive enzymes. However, it has its own drawback in the sense that the capsule can rupture, or there could be a leakage. And sometimes we have a complex fabrication processes which can make it to be more difficult. The use of encapsulation and entrapment is that among the immobilization method, it is applicable to plant cells and is the most common and effective method for solid support in application in enzymes or from plants or plant products, because the enzyme is trapped in a porous or gel matrix. Example, we have agarose or carrageenan. The next is covalent binding technique. The covalent binding immobilizes biomolecule by forming strong chemical bonds with the support materials. This ensures they remain fixed during use. The tight attachment enhances thermal stability but restrict enzyme mobility, and which can reduce their activity. So because these enzymes are anchored on surfaces, surface contact remain efficient. However, the method is less suitable for cell immobilization, and the support materials are typically non-renewable. So the technique is less effective for immobilization cell, and support materials are non-renewable, like I said. Next one is cross-linking method. The cross-linking method is a powerful technique that blends covalent bonding and entrapment, creating a stable matrix that holds enzyme in place. So the cross-linking agents such as glutaraldehyde form chemical bridge between enzyme molecule or between enzyme and the support matrix. They can react with functional groups such as amino group on the surface enzyme, creating a network that traps the enzyme in a fixed position. Advantage of this is that there is thermal stability and reduced enzyme loss during repeated use, and is suitable for creating carrier-free enzyme aggregate which are compact and efficient. The limitations is that it may reduce enzyme activity if the structure becomes too rigid, and most of the time the structure can actually become too rigid and require careful optimization of cross-linker concentration and reaction condition when using. In procedure for enzyme immobilization using covalent bonding and cross-linking process, the procedure for enzyme immobilization technique involve, the first thing is selection of enzyme. For selecting enzyme, an individual choose an enzyme that is based on his desired process. Different type of enzyme can be used, but you have to select your enzyme, and these enzymes are selected based on their stability, compatibility with the immobilization method to be used. After the selection, then we have to also select the immobilization matrix, such as agarose, cellulose, chitosan, or silica. These are immobilization matrix that are suitable for immobilization technique. After selecting this immobilization technique, solid support, also we can also select magnetic beads. They are also selected based on their porosity, surface chemistry, their mechanical strength, and even their enzyme compatibility with their choice of enzyme. Remember that you have to select your enzyme based on what you want to do. And the enzyme being selected also will determine the support. Then the next step is the preparation of enzyme mixture. The enzyme is now dissolved in the appropriate quantity. The appropriate quantity of enzyme is dissolved in the appropriate quantity of buffer solution. That is, there is a measurement of the enzyme that must be dissolved in a buffer solution. Example, we have a phosphate buffer which is used, and we maintain the pH and temperature for the enzyme. Each enzyme has their own optimal temperature for their stability. After that, then the enzyme and the matrix is now activated. Activation of the matrix is treating the matrix with an activating agent to introduce the reactive groups. So we have examples that are here. We have the glutaraldehyde, and we also have the epichlorohydrin and carbodiamides. The next step is enzyme binding immobilization. In this enzyme binding immobilization, the enzyme solution is mixed with the activated matrix, and then it is now incubated under optimum temperature condition and pH to allow a covalent attachment together. Then we go to the next step, which is the enzyme cross-linking, which is the stabilization. At this process, a cross-linking agent is added to reinforce the enzyme matrix complex. And this cross-linking agent usually enhance thermal and operational stability of the immobilized enzyme. Then we go to washing, we wash it and store the enzyme until use. You can remove the unbound enzyme by washing in a buffer solution, and the final product is now stored in a desiccator until the use is now required. So this is the step or procedure of a small scale enzyme immobilization procedure. But at industrial scale, these procedures are more elaborate. But in the laboratory, this is the step that is used in enzyme immobilization procedure. These are the advantages and disadvantages of each technique. At your free time, you can go through the advantages and disadvantages and compare and contrast these advantages. So without much ado, we go to application of this process. The first application of immobilization of enzyme is in the use of the process in the dairy industry. In the dairy industry, we use the immobilization of enzyme technique in producing dairy products. Example is production of lactose-free milk. The immobilization technique is used in dairy industry for immobilized lactase enzyme, which are used to produce lactose-free milk, especially important for people who are lactose intolerant. They lack enough of this natural enzyme in their system, such as lactase. So in their digestive system, when they consume milk that contain this lactose, they are unable to break it down, and it causes discomfort like bloating or cramps. And to solve this process, the milk is passed through a column containing alginate beads. The alginate beads is a type of beads that is a support for entrapment of enzyme, which allow the immobilized enzyme lactase to break down the lactose. So as the milk flow through, the enzyme breaks down the lactose present in the milk into glucose and galactose, which is then easier for them to digest. So that when they consume this milk, they can now enjoy the milk comfortably. For people who do not have any problem with lactose, for the lactose tolerant individual, this process is just not necessary. However, they can consume the milk without issues. But then, for lactose intolerant population, this enzyme-based method makes milk safe and comfortable for them. So the process is very important. The next application is in the juice clarification using immobilized enzyme. Also, in fruit juice production, pectin is a natural substance which is found in fruit juice. It makes juice cloudy and thick. However, some people prefer their milk to be clear, and they prefer their juice to be clear, and this juice must look clear and smooth. But the use of pectin normally makes this to be cloudy. So this is a problem that has to be solved. So to solve this problem, the juice is passed through a column. This diagram is just a simplified diagram on how this looks like although in the industrial scale, it is more complex. However, to simplify the process, for you to understand, this diagram is just a simplified diagram, like I said. So the juice flows through the enzyme, break down the pectin, making the juice clearer and easier to filter. And as the juice is collected, a clearer juice is collected, and now everybody can enjoy it. So the process is useful in commercial production of juice because the enzyme remain intact and can be reused again and again. And the juice doesn't get contaminated with the enzyme residue because it's only break down what needs to be break down. It clarifies the juice, and it improves their appearance, and also the shelf life and consistency of the final product is intact. Just like lactase, it helps lactose intolerant people enjoy milk. Pectins also help juice lovers enjoy a cleaner and more refined juice in drinking it. Now we also have the application of this in pharmaceutical industry in synthesis of active pharmaceutical ingredient. The diagram here shows a substrate that flows through a column path, also a simplified diagram, in which immobilized enzyme result in the production of active pharmaceutical ingredient. Now, like I said, the diagram is a streamlined process of synthesizing active pharmaceutical ingredient (API) using immobilized enzyme. The technique allows precision and efficiency in drug manufacturing. At the top is the substrate. Essentially, the raw chemical that is input is introduced into the system and directly downward into a container packed with beads. These beads again contain the enzymes, and these beads are not just filters as previously known for the lactose and also for the juice clarification. They carry immobilized enzyme that allow them to act as biological catalysts. As the substrate flows through this enzyme-rich environment, it undergoes a targeted chemical transformation, and the enzyme being immobilized remains fixed in place and can be reused again multiple times, which reduce waste and improve consistency. So at the bottom of the setup, the transformed product emerges as the desired API, that is the active pharmaceutical ingredient, ready for further refinement or formulation. So this method is particularly valuable in pharmaceutical production because it allows for controlled reaction, minimize contamination, and support scalable, cost-effective synthesis. Lastly, for sweetener production, in terms of sweetener production using immobilized enzyme, in industrial setup again, a glucose-rich solution, usually from cornstarch, is passed through a packed bed reactor filled with immobilized glucose isomerase enzyme. As the solution flows through, the enzyme converts glucose into fructose, resulting into high fructose corn syrup, a more sweeter and more desirable product used in soft drink and processed food. So the mobilized enzymes stay fixed as usual, allowing the process to run continuously and efficiently. So as you can see from the diagram, we have the HFCS, that is the high fructose corn syrup, being collected from the other end, and the reactor maintains the optimal condition, and then the production continues. So these are applications of the immobilization technique. There are so many applications of immobilization technique. These few are just being discussed, and I believe on your personal study you can go ahead and see more. So immobilization techniques refer to the confinement of, in conclusion, it refers to confinement of enzyme molecule onto or within a solid support, which enables their reuse and enhance their operational stability in biochemical processes. The technique is widely employed in industrial application, like I said previously, in sweetener production. Then we also have in biofuel synthesis that I mentioned, also in wastewater treatment. So this process offers several advantages over natural enzymes. And they have improved thermal stability, pH stability, and they reduce contamination of the final product, and the ability to operate in a continuous flow system, and they are reusable methods. Their usable ability. allow them to be you know, available for industrial process, and from the previous method of industrial process, this advancement have allowed more faster production of industrial scale production of several products as being applied respectively. So this technique, as discussed, is very wonderful and useful for industrial microbiology. And I hope at the end of this lecture, you have been able to, see the importance of immobilization technique of enzyme. Thank you very much and have a wonderful day.